Hey everyone, thanks for making it today to this very impromptu fireside chat. We're really excited to start sharing our results, and we have a pretty wide team from AgelessRx here today. Girish, Dr. C, and our VP of Applied Sciences, Stefanie Morgan, who they'll introduce themselves in a second. So, Girish, maybe pass it on to you.
Yeah, absolutely. Thanks for that, Mauricio. So super excited to dive into this. I've been in the longevity field since the Hallmarks of Aging paper came out, enamored with the biology and, dusting off that translational bridge between the preclinical data and human longevity. Yeah, joined as a senior research manager of the Applied Science Department. Must have been early 2023, and it's been a great ride ever since. So excited to dive in to the details of one of the most promising longevity drugs in existence today, rapamycin. From there, I'll pass it to you, Stephanie.
Yeah, I'm really excited to be part of the team. And I got here, joined the project when all the data was done, collected, and now we just had the fun of analyzing it. So it's been really cool to see all the efforts of the hard work and take an objective look at that. So I'm really excited to share it with you all today, and can't wait to hear some of your questions.
Dr. Z.
All right. So I think some of you know me already. My name is Zelzla, Dr. Z for short, co-founder and chief medical officer of AgelessRx. We kind of started down the rapamycin research path almost from the very first day that we launched AgelessRx back almost it'll be 5 years going on October. So yeah, very excited. I think I'm more excited now about rapamycin than I have ever been. Just the promise of it, and a lot of the a lot of interesting data coming out, a lot of interesting studies that have been launched, and a lot of interesting anecdotal reports as well. So I'm really looking forward to continuing to expand the body of research for longevity for rapamycin for human longevity.
Awesome. I think then we can, dive into the presentation and,
Sure.
just a reminder, if anyone has any questions, feel free to drop them in the chat. We'll be keeping track of them and keeping a queue. And then at the end of the discussion, we'll start, getting to them one by one. So with that, Dr. Z, I think I'll let you start off and take it away.
Yeah. You want to move to the next slide? There we go. All right. So today we're going to be talking about we're going to be talking about the bioavailability paper it's been submitted for publication, also submitted to the preprint servers. Obviously publication takes a few weeks, a couple months sometimes. But the preprint server paper should be coming out. We were hoping it'd be out by now, but it'll be out very shortly, and we can kind of post links to that once it actually comes out. But we'll be talking about the data today. We'll be talking a little bit about rapamycin, but I just want to kind of set the stage that this is kind of a prelude or kind of a intro call to our larger Pearl clinical trial. So today's talk will be talking about the bioavailability. We're not going to be going over much of the Pearl data. So that's a different call scheduled August 27th at 1 p.m. and invitations will go out as soon as we kind of lock down all the logistics of it. But it's tentatively scheduled August 27th. So today we're going to be talking about introduction. We've already made some introduction to ourselves, background and rationale of why we kind of commissioned this study, what are the key findings and implications of the findings, future steps, and then we'll kind of open it up to the community to field some questions and answers, and try to get to as many questions as we can, respecting everybody's time. Next slide. Is my audio okay? Audio check is good? All right, good. All right, so we've kind of already made intros here, so you can skip that slide. All right, so a lot of people know about biology of aging and things like that. Girish, do we have a slide about kind of the background of the study itself, or is that further down the line? Oh, that's further down the line. Okay.
Yeah.
So Girish, why don't you go ahead and take away the intro to like why is it important that we're studying this, and then I can jump into the background of the study itself.
Yeah, sure, absolutely. So if I can go backwards here. All right. So I think, as all of us in this room are probably well aware of, why targeting the biology of aging is so key to longevity. Biological aging is this real process, this undercurrent of cellular and molecular damages and dysfunctions that occur in the body that eventually give rise to the waves of disease and frailty later on in life. And this is something that within the conventional, healthcare system, individuals aren't paying attention to these subtle changes that occur for decades and decades and decades. And we're really only honing in once pathology already arises, when complexity has already taken its form and it's a little bit harder to address the root cause of disease, which is this biology of aging. And that's what makes it the biggest risk factor for chronic disease and mortality. So it's really the rate of biological aging that determines our lifespan, how long we live on Earth, right? The number of candles on our birthday cake, as well as our health span, which is equally, if not more important, in that this is the time period that we're living in good health, with high quality of life, free of disease and frailty. And by enhancing our health span, we have the opportunity to not only live fuller, high quality lives, but also extend lifespan as a side effect, right? If you're healthier for longer, presumably you can live longer as well. So that's really center of the bullseye for the field right now, as we, dive further into discovering these promising interventions. So yes, one of the three rules of the biology of aging is, it's occurring under the current cellular damage and dysfunction, occurring over decades, and it is modifiable. And we see that within the lab. And now the big thing is translating that within human studies. And there's a lot of there's a lot of potential there. Um, so, how can we do that? Of course, there's lifestyle change. There's things that our, grandma told us about diet and exercise and who we surround ourselves with. But then there's also a more targeted approach, which is using gerotherapeutics, right? Interventions that directly target molecular and cellular mediators of the biological aging rate. Some of these we're well aware of, like sirtuins and AMPK. And for the purposes of this presentation, mTOR really becomes center focus as this major regulator of cellular homeostasis and autophagy and growth. And it turns out, well, we'll get to that in a further slide, but it turns out that we can use drugs in a very directed way to modulate mTOR so that it operates in more youthful levels as we age. And by doing so, we have the opportunity to continue optimizing health throughout an individual's lifespan, prevent multimorbidity, and extend lifespan and health span. I can go into the background of rapamycin as well, Dr. Z, and then lead into or sorry, you're on mute.
Sorry, I'm muted. Yeah, maybe just a few minutes. I'm
Okay.
assuming most people on here are kind of already well aware of
Yep.
rapamycin. So maybe just a quick overview just for the sake of completeness.
Sure. So rapamycin slows biological aging through inhibiting, mTORC1. There's an mTORC1 complex and an mTORC2. The hypothesis within the field right now is that more selective inhibition of mTORC1 is what leads to longevity benefits, while inhibition of mTORC2, which, they're distributed differently within different organs and tissues, inhibiting mTORC2 is a little bit sloppier, in which it might lead to some of these metabolic consequences that you may see. So there's a lot of efforts now, in terms of designing very selective drugs to target mTORC1 and extend lifespan and health span. And again, a lot of this hypothesis is formed within laboratory studies, and we're really sussing that out now, within human data. So why is rapamycin so promising? Well, it's been shown to extend the lifespan in every organism it's been tested on across billions of years of evolution, from, unicellular yeast and all the way up to non-human primates like marmosets. So it's been able to mitigate or reverse age-related decline in many organ systems. And I emphasize reverse because there's actually been shown of reversal of periodontal disease and aspects of immune aging as well, which is extremely promising. Joan Manick ran some studies, and I think these are the most robust studies we have to date on everolimus, showing that it boosts immune health in healthy elderly individuals, specifically by boosting antibody response to influenza vaccinations. But, where do we really stand? How do we apply rapamycin more broadly and effectively within healthy longevity medicine to realize its true potential, right? It could be much more than that. And that's what we're really focused on here. I think in terms of the optimal therapeutic dose of rapamycin, it's not yet established, right? There's a wide range of dosages that individuals are engaging with. Krister, who's on this call as well, has some great images of this in which he interviews longevity leaders, and everyone's taking somewhere between 1 milligram per day to 20 milligrams biweekly. And there seems to be some consensus on 6 milligrams per week, largely driven by early studies by Joan Manick and actions by Alan Green, who's a clinician that's prescribed to a bunch of individuals interested in rapamycin, but we still don't have the details of what's going on, in terms of pharmacokinetics, in terms of biology in normative aging, healthy individuals, right? A lot of our data is within diseased populations, organ transplant patients and cancer patients. So this is a really important early times to discover what that, right therapeutic dose and therapeutic blood levels are for rapamycin.
Yeah, and this slide comes from, I believe the slide is from that survey, right? That registry survey. Yes. The 333 rapamycin users that were surveyed. Okay. For anybody wants more detail about that study and that survey, we did a webinar with Matt Gaberlin a few months ago. It's recorded and posted on our YouTube channel at AgelessRx.
Yeah, that's a great call out, Dr. Z. And these were individuals like a real world study taking rapamycin anywhere from, a few months to several years, relatively clean safety and trends of decreased severity of COVID symptoms and, a bunch of quality of life parameters improved. So really promising early data there. Let's get into this. Okay. So, again, still many questions remain regarding rapamycin dosing for healthy longevity. Within, generally healthy individuals, how much rapamycin is getting in the body? Do we really need to be digging into this, or can we just continue taking rapamycin and seeing how it pans out, in terms of clinical effectiveness? Conventionally, the way to do it is to really track it down to blood levels and see what you're doing in the body, rather than taking it by faith that what's happening in organ transplant patients is the same thing that's happening within us as well. And this is just like prelude to one of the big impacts that this rapamycin bioavailability study that we ran is making. How do we achieve therapeutic blood levels of rapamycin for healthy longevity? What formulation is the best? And I think Dr. Z can talk about the background from here in terms of, our activities around determining the value and the challenges with compounded rapamycin versus generic rapamycin. There's a lot of, I think, conflicting views within the field. I think primarily because there's not that much data published on compounded rapamycin and standardized data. And I think this is, a real take-home point that we want to bring home from this presentation is we really, dug into this with as much rigor as we could within the context of a real-world study, and really excited to dig into it further. Dr. Z, if you want to go into the background, I think this is
yeah. So for those of you who've been following kind of our progress on the Pearl trial, I think I'm sure at least some of you kind of have followed us on our previous webinars for Pearl, probably dating back, 2020, I think is when it was some of our earliest webinars on Pearl came out, talking about the project and, the study design and whatnot. Just like any scientific study, especially one where there were so many unknowns, there was bound to be changes and things like that. So again, I don't want to like spend too much time on the Pearl trial because we'll be dedicating a full webinar to that coming up in a couple of weeks here, in about 10 days. But But it's important to kind of highlight this data separately because again, I didn't want to like dominate the Pearl webinar with this data either. So kind of works both ways. But just a brief background is, going back to 2019, metformin was thought to be like the king of longevity, among the longevity community. And people had just started getting interested in metformin, for example. But then there was this new kid on the block called rapamycin, that was even more promising according to a variety of different data points that Girish talked about. However, metformin was like very well known. I mean, like we know like thousands and thousands of studies, we knew it kind of like upside and down, and we know all side effects, the pharmacokinetics, like we know pretty much almost everything there is to know about metformin, although it still does surprise us. But rapamycin was not the case with rapamycin. The data was very limited to, Girish talked about, a few small scale human trials and a lot of animal trials that were hard to extrapolate. So that's why we launched the Pearl trial back. We started it back in 2019, and then the pandemic hit. Slowed us down a little bit. All work halted on research during the for the first 6 months of the pandemic, and then we finally picked it up and launched it again. But the reason that's important is because when we were designing the arms, the intervention arms for the Pearl trial, we had hired some researchers, and we had consulted with clinicians and things like that, as many people as we could talk to in the field at the time. We had some various different recommendations on dosing. I wanted to make the trial to be as practically applicable as possible moving forward. And so that's why I came up with the idea of using the compounded version of rapamycin, because at the time that seemed to be the one that was favored by some of the clinics that were prescribing rapamycin for longevity. At least that was my impression at the time. And there's also other considerations. I think the biggest one that a lot of people don't appreciate is this last ring on here, the placebo design. When we decided we wanted to make the Pearl trial a randomized blinded trial, the shape and the shape of the commercially available tablets presented a big challenge. Also, the dose limitations, as many of sirolimus as a commercially available tablet, whether it's the brand name or generic, only comes as 0.5, 1, and 2. So if we wanted to test different doses, it automatically meant we would have to do multiple of tablets. And then all of a sudden now you're talking about 2 placebo arms, and then so you can stay blinded, for example, and then also you'd have to find a placebo that was identical in every way to the commercially available. And when we looked at it at the time, it was just not feasible. And then and then it turns out somebody who is doing a randomized controlled trial right now, better funded than we were. It turns out it is a very extremely expensive. I don't quote me on this, but I think I heard that the placebo that the company producing placebos for this trial that I'm referring to, it was quoted like $50 a tablet for placebos. So I mean, and then if we want to do like the standard 6 milligrams, for example, that would have been a minimum of 3 tablets per week. So the cost would have like ballooned out of control had we had gone down the route of using the commercially available tablets. And like I said, I always envisioned using the compounded because of the dosing schedule. Compounded almost always provides more flexible dosing. I come from a background of using low-dose naltrexone, and low-dose naltrexone is compounded, and I really like the flexibility of the compounded LDN, which is another topic for another day. And then cost. We discovered that the commercially available sirolimus, if you look on GoodRx, for example, I think the average cost for 12 tablets is $90. And that's for the generic. If you want to go to Rapamune, it's like $3,000 for 12 tablets, which is ridiculous. And the other thing that's not on here is currently there's only 2 generic makers. There's Dr. Reedy, and then I forget what the other one is. I always forget the name of the other one because we don't currently use it. But if anybody who knows anything about the pharmaceutical industry, generic medications are always at risk of having some kind of supply disruption. Or whether it's some kind of contamination issue, like we've seen with like, ranitidine and, other, blood pressure medications and things like that. Or a lot of times generic manufacturers they say, you know what, this is not a profitable drug. We're gonna yank it for the market. So there's been many cases in over the last 15, 20 years where generic manufacturers, they just said, okay, we're gonna stop making it, or they have trouble. And so again, my thought at the time was the compounded would provide a much better option for the community. So we're not at necessarily the mercy of these generic manufacturers. And I think, it's funny because I think sirolimus for longevity, I think has greatly eclipsed the prescriptions for sirolimus for transplant, and so I think the off-label use has eclipsed the by far, I think, the use. At least that's the rumor has it. I don't have any hard data, but that's what I'm hearing from various different sources. And it makes sense. I mean, the survey we did, 333 people, but those are only the people we were able to get to fill out the survey. There's probably at least, one estimate, 10,000 people taking sirolimus off-label in the United States. And so there's a lot of pressure from I think the generic manufacturers. So I know I'm ranting on about that, but I just wanted to put a very, I just wanted to make like a very clear rationale for why we chose compounded rapamycin for our trial. Now, what we didn't know at the time, and it didn't come up with any of the experts or was the bioavailability issue. So when we, like I said, when we dug into the data, everybody that I spoke to was under the assumption that all the pre-FDA approval trials and a lot of the animal trials and the cell culture trials, they were all based on just what you buy from like Sigma Ultra, Ultrich or something, kind of a bulk generic or bulk manufactured molecule. And so at the time, I didn't think it was an issue to use compounded because I assumed at the time, like a lot of other people did, that that this is what was used in the studies, and this must just be just fine for clinical use. And like I said, there were at the time a lot of clinicians, I mean not a lot, but there were a few clinicians using the compounded version at the time as well. However, about I think a third of the way throughout the enrollment. So we got about a third of the way through the enrollment of our study. And then it was brought to our attention that Pfizer, who holds a patent for Rapamycin, had come out with an encapsulated form of sirolimus to enhance absorption. And then it was also found that during the ITP study, and I think this is kind of where a lot of the misconception with the rapamycin, compound rapamycin comes into play. With the ITP study, they had tried using the bulk, rapamycin for the mouse trial, and they couldn't, mixing it with the feed. Apparently, they couldn't get mice to eat enough food or to absorb enough rapamycin to be found in the blood, and so it was kind of it was fortunate and unfortunate. The unfortunate thing was it delayed their trial, but that's also the fortunate thing because that kind of led to them then starting the trial. If anybody knows the history of that trial, starting the trial in much later, much older mice and actually finding a benefit there. So that was actually one of those things in science where it's like, they would have never predicted that, or they would have never like designed the study that way, but because of this. And I can talk a little bit about like my opinions of why the mouse study didn't, for ITP kind of failed to show blood levels. But in humans we do see absorption, and again that's why it was important to do this trial. So we did it for a couple reasons. One was to like figure out like, okay, and we know the encapsulated, commercially available stuff that's absorbed because, for people who undergo transplant, for example, they have to check their trough levels as part of their, to keep within that therapeutic window, et cetera, et cetera. But we didn't know so much about how much the compounded was. And then nobody really knew what the difference in the bioavailability or the concentration was between the two formulations. And like I said, we had kind of already committed to use the compounded version during our Pearl trial. So it was data that we really needed before we could, before we published our Pearl trial, we needed the data in order to kind of like help, discuss some of the potential limitations of the trial as well. And also, like I said, it all comes back to practicality. Like what did I want to, or what did I want people to be prescribed? Like I said, I really thought that compounded would be kind of the way of the future. And I think it still is. Hopefully after today, most people will agree that it is potentially a very good source to choose from for the compounded. So the other questions that are outstanding, if you go back for a second, Girish, can you go back? Oh, yep, there we go. There we go. So the other was like, what's the right dose for an individual person? And then how to achieve therapeutic blood levels again, in an individual person. The only thing I'd like to add to what Girish was talking about is conceptually, there should be a relationship between somebody's sirolimus levels and their outcomes. But that may or may not be a correct assumption. It's a starting point, but I think it's entirely possible, and this is just my opinion, but it's entirely possible that even the same therapeutic blood levels will have different effects on different people. So, and again, I mean, but we got to start somewhere. So first we're starting with the sirolimus levels. And then once we figure out like what are the measurements that we look at, whether it's like mTOR activity, autophagy activity, other markers, and that's kind of something, maybe we'll talk about a little bit as we kind of get toward the end of the presentation. But I wanted to highlight that difference that, by no means are we suggesting that understanding sirolimus levels in and of itself is going to give us the answers we're looking for. But it is a step in the right direction. On to the next slide. So a little background on the Pearl trial that I forgot to mention is when we designed the trial, we actually were going to include the sirolimus levels in with the participants, and we did that for the first few participants, and then the way, I think we were using Quest Labs. The way Quest Labs work, you can go online and get your own blood test results. There's no way for us to like shield people from the results. So we realized where people were going online, we had very sophisticated participants, and thank you, for those people in the group that are participants, we had very sophisticated participants, and they knew they could kind of like try to figure out whether they're on the placebo group or not by checking their Quest Labs and seeing whether they had sirolimus in their blood. And so once we realized that, we had to stop checking sirolimus levels. We had to modify our protocol and stop checking sirolimus levels on people. So we didn't end up checking sirolimus levels on people until after they completed the Pearl trial. Otherwise, it threatened our blinding. And so like I said, we had to stop doing it. So that's kind of a lesson to be learned when designing these kind of clinical trials. Is you got to be very careful on what you test people, what you give people. Otherwise, it could potentially threaten your blinding. So the fortunate part is many people who were in the Pearl, once they finished the Pearl, were then also included in this data. Blood draw. So what we did was once people finished Pearl, we would kind of roll them over into our telemedicine platform for those who wanted to continue taking rapamycin, and those who were in the placebo group were offered rapamycin as well. And then we also eventually opened up a rapamycin offering to the general public, kind of a limited pilot offering, but now it's available to all patients who are interested in it. But at the time, we kind of opened a limited pilot. So then we decided to enlist those patients into a voluntary blood draw, where we would ask people, hey, you're taking rapamycin. Do you mind going for blood work? We want you to do it 24 hours after you ingest your rapamycin, and we were fortunate enough to have a lot of people end up taking us up on that offer and allowed us to make this publication happen. In terms of why 24 hours, why not like peak? So a couple things about peak. It's very hard to get. I think I would say it's actually in the clinical setting, it's impossible to capture the peak. Rapamycin peaks between like 2 and 4 hours after ingestion, and it's actually quite a high peak. So it peaks really high, and then there's a dip, and then in our paper we talk a little bit about kind of a second peak, because it's taken up by the red blood cells. And then, once it reaches a certain level, the red blood cells start giving it off again. So you get a small little peak again. So that, so a lot of people say, why didn't you check peak levels? But peak levels in the clinical setting are impossible because it spikes quite high. And so you don't know whether somebody's peak is it at 120 minutes? Is it 125 minutes? Is it at, 95 minutes? And so I think it would have been a fool's errand to try to figure out to try to catch the peak, which is why we didn't. We had listed some volunteers and we did different time measurements before this. It's kind of like a run up to this study. And we had decided that 24 hours after ingestion was not only more practically applicable, instead of, let's say, 20 hours, or instead of like, I don't know, 26 hours, 24 hours, just like, it makes sense. You take your pill at 8 o'clock on a Monday and you go 8 o'clock on a Tuesday to get your blood drawn. So it was a kind of a balance of practicality and also science. And there's a lot of indicators that 24 hours is the right measurement. Girish, can you talk a little bit more about that? I know you were the one who kind of dug kind of deep in that whole 24-hour level.
Yeah, I mean, just as a real quick, a lot of the trough levels that are measured within organ transplant patients, a lot of the PK data does correlate with that 24-hour time point just because of daily dosing those individuals. So it's, fairly robust. And again, just like, double clicking on that point that you made about peak levels, is extremely hard to catch and extremely variable because of the initial biology in which, how much you're going to see at peak depends on how much rapamycin is going to insert itself in fatty cell membranes and, the particular variant of cytochrome enzymes that are in the body that actually metabolize rapamycin. So that's different for different ethnicities, for different types of people. So you're just getting a much dirtier readout in terms of getting reliable standardized levels. So I think there's a lot to say on that level about that 24-hour measurement when you actually give the body some time to actually metabolize it and let those levels stabilize a bit.
Yeah, so it wasn't, it's not too far out, like, for example, if we would waited, 5 days, for example, there may not be any sirolimus. And like I said, if we did it too close, then that presents a practicality issues as well as it introduces a lot of noise into the data. So 24 hours seems to be a good, again, that's one of the limitations of the study. It's only 24-hour levels that we've included here. So it's possible, some of the conclusions might be kind of varied or skewed if you look at different time points. But I guess the point is to compare the 24-hour levels between the commercially available and the compounded, and kind of like the major point here is dispel the myth that compound, like the non-encapsulated, the compounded, does not get absorbed. So I think we can put that. I think today we can put that to rest. All right. So what are the key findings today? Girish, you want to, or Stefanie, either of you, if you want to kind of go over these?
Yeah, I mean, I could start it off, and then perhaps we can discuss it. But this is just like the baseline information demographic. This is the percentage of our study cohort that took either compounded or generic rapamycin. You can see more people included that took generic rapamycin. These are the different dosages. Just pointing out very few people that took 8 milligrams and of generic and 5 milligrams of compounded. So because of the nature of the real world study where we're sending out this request to individuals to contribute their data, we're casting the net broader, and that's great because we're collecting more data. But then it becomes a little bit more expansive, less narrow, because we're getting data where we can get it from, right? So that's why we have this type of distribution of people taking different dosages, and then we can compare what the blood levels look like across dosages for people taking either compounded or generic rapamycin. Just to kind of double click on this point, at the start, there were not significant differences between the compounded groups and the generic groups in BMI or gender. You can see that there's more of a skew towards men within both groups. And, I mean, just take a look at the BMI was pretty optimal for the participants, again, emphasizing the fact that, between 23, 25, emphasizing the fact that these were healthy individuals taking rapamycin. Where we did see a difference between the cohorts from the start is that individuals taking compounded rapamycin were slightly older. I believe the mean age was around 61 versus 57, somewhere around there. So that's an important piece of information to take into account when going into the study. Here's like high level point number one. And, I'll also open the floor up to Dr. Z or Stefanie if you want to double click on anything about any of these key results. But right off the bat, you could see, right, you could see the orange dots and the diamonds as the generic dosage. We broke it up because we had individuals that would opt into getting blood tests. Within the context of this real world study, we had some people opt in for 2 blood tests, and we're not going to just throw out that data. We're going to use it. A lot of our statistical analyses, though, were done on people's most recent dose. I just wanted to emphasize that point. But you could see the dosage distribution, and the blood level distribution of people taking generic rapamycin and compounded rapamycin in gray here. And overall, you see people taking lower dosages of generic rapamycin, either 2, 4, 6, or 8. And they're getting, it's more potent. They're getting higher blood levels of sirolimus or rapamycin after 24 hours versus individuals taking 5, 10, or 15 milligrams of compounded. One thing I want to point out, though, is rapamycin levels were detectable across dosages, across formulations. But rapamycin is bioavailable in both formulations after 24 hours. That's an important point. But one thing to emphasize is that the generic rapamycin had a steeper increase of dose to blood sirolimus relationship. As you can see, as you increase the dose, you're getting a steeper increase in blood levels. And with compounded, it's a bit flatter. There is an increase, but it is flatter than the generic.
So one thing that people might ask is like, why the different dosings? So I forgot to mention that. So our protocol at AgelessRx is to start people low. So like, if somebody selects the commercially available generic version, we start them at 2 milligrams per week for the first month, and then 4, and then 6. And then, if somebody wants to try a higher dose, we can go up to a maximum of 8. So that's why we had like 1 or 2 people on 8, because, they wanted to try the higher dose. So that's why we have different doses. So we kind of like, if we caught people when they were titrating, that's why they're at 2 and 4. And then, 6 was kind of the continuation dose. Likewise with the, with the generic, with the compounded, it was 5, 10, and then 15. So that's why we have different dosages here. But it also gave us an opportunity to test people. I don't want to steal thunder, but also to test people as they went up on their dose. And we have a few data points to present on that as well.
Yeah. And, because we had individuals within the compounded groups and generic groups taking different dosages, right? Just because of the nature of the titration schedule for each formulation, we attempted to get a relative assess the relative relationship in bioavailability of generic and compounded by normalizing it by each milligram of rapamycin taken, right? So if you're taking two milligrams, then whatever your blood levels are, they're normalized by each milligram of rapamycin you're taking versus someone who's taking 15 milligrams, right? So this is a way to say, okay, for each milligram of rapamycin I'm taking, this is what my blood levels look like. So you can compare across groups, right? It's a very crude way of doing it, but it's the best sort of strategy that we came up with the real world data that we had in mind. So one interesting point here is that, when you normalize the blood rapamycin levels per milligram of rapamycin taken, you see that as you increase the dosage of generic, generic formulation, you see a sort of decrease of bioavailability, right? So this is suggesting perhaps some sort of saturation going on in terms of, when you're taking the 2 milligram dose, you might be absorbing 80% of that rapamycin, but then as you increase the dosage, you're absorbing less and less per milligram of rapamycin you're taking. And this effect isn't as stark within the compounded group, suggesting that, when you're taking compounded rapamycin, you're absorbing relatively the same amount of rapamycin per milligram. So that means that when you're titrating up the dosage, you can more easily predict, how much rapamycin you're going to get with a formula, right? You could say that if I'm getting 25% of bioavailability with 5 milligrams, I can expect something similar with 10 milligrams, while there seems to be a decline in bioavailability as you increase the dose of generic, right? And we can talk more about that, but I just wanted to point out, I know we have a slide on this, just pay attention to the, huge variability in response, right, to the same dose of rapamycin, whether you're talking about generic or compound formulations. I think that's another key point here. And, if you look at the relative bioavailability of compounded versus generic rapamycin, as Dr. Z had mentioned before, we see a 1/3 potency relative 1/3 potency of compounded, compared to generic. And this is important data to keep in mind as we think about prescribing compounded rapamycin. It's not that it's not bioavailable, as many, the ITP study and other, leaders within the field have proposed. It's that, if you get it from a quality source, you have this 1/3 relationship, more or less. With all the differences in bioavailability, right? You have a 1/3 bioavailability when you compare compounded to generic, but given the dosages, we got relatively similar blood levels of rapamycin within the blood, right? Whether you're using compounded or generic. Yes, we were using smaller dosages of generic and higher dosages of compounded, but they still resulted in the main goal of, eliciting similar levels of rapamycin within the blood, indicating that we can, there is some comparable efficacy in terms of bioavailability if we just get the compounded dose high enough. And I think that's an extremely important point. Most people end up between 1 to 4 nanograms per ml of rapamycin after 24 hours. Part of our review part of the paper that's going to be published soon in Giroscience and should be available in Med Archive soon is that, these dosages within 1 to 4 nanograms per milliliter after 24 hours seems to converge with efficacy data for other trials, right? In correspondence with Joan Manick and the small pilot trial that we had talked about before, she actually saw similar levels of everolimus, in this case, within the blood after 24 hours. It was, a Cmax. So she used a 0.5 milligram dose of everolimus and a 5 milligram dose of everolimus, and she saw similar blood levels after 24 hours within that 1 to 4 nanogram per ml range, and saw these immune benefits with it. So you can think about how you can reach that with either formulation, which I think is another sort of check in the box of compounded rapamycin.
Yeah, well, actually, one thing I forgot to mention, and I hinted to it, but we went into this study with with much smaller data set, hinting at this 1/3 relationship. So we kind of knew up front, and this study was just kind of to confirm it with a larger data set. And we have a larger data set yet that we haven't even extracted, but I have no idea when we're going to extract that and analyze it. But just to let you know, I mean, we knew kind of going into the study based on a small data set, like I think 3 or 4 volunteers. So that's why we were able to kind of like use that 3-to-1 ratio when it came to dosing.
I'm going to try to get through the results a little bit quicker so we can get to some of the good questions that we see pointed out here. But, what we see here, as I mentioned, there's some pretty significant inter-individual heterogeneity for 2 people taking the same dose, 2 to 6x difference of, two people taking the same dose of generic or compounded rapamycin, just emphasizing the point that measuring sirolimus levels or blood rapamycin levels is actually quite important if you really want to see what's going on in the body. Is this the right dose for person? Are they even, absorbing something when you're taking a 5 milligram dose, or do you need to titrate up? Because you can kind of waste a lot of time, and people might not be experiencing clinical benefits, and part of that is likely explained by the differences in bioavailability from two different people taking the same exact dose across formulations. This is whether it's generic or compounded. So we did not see, when we really dug into this, we did not see any of these effects explained by BMI or gender. You can see here when the rapamycin was normalized per milligram rapamycin taken, there weren't any significant differences when we stratified by BMI and by gender. And this is in opposition to some of the other research that's been shown within animal studies, or preclinical studies, in which, in the ITP studies, there was a female bias in terms of bioavailability, and within, Adam Salmon's marmoset studies, there was a male bias in bioavailability. We did not really see that with our study here.
That's good news for humans. It means that we can study we can use the same dose and kind of study it across both men and women.
Yeah, and then I don't know if I have a slide on this, but like Stefanie really dug into it, and we looked at she looked at all these factors from age and time on dose and just a number of others, and we really didn't see any differences from these different background characteristics influencing bioavailability. So
yeah, I mean that's a whole other discussion. Like we could have a whole webinar talking about how like animal models, because of this huge sex difference in other animals that usually don't even translate in humans, why we always have to take animal models with a grain of salt.
Yeah, anything just to kind of add on to what Girish said, we looked for every obvious explainer that we could find. And the one thing we didn't measure in this study was like the microbiome or the gut health or real fine-grained dietary considerations. And we think those are likely quite important because we looked at pretty much everything else and it didn't seem to have a relationship that we could detect.
Definitely something to study in the future. Right. So let's finish this off so we can get the Q&A.
Kind of these are two ancillary slides. We did have the opportunity to do, since individuals, we had participants taking, two dosages across time and showing us their blood levels or relaying that data to us, is, we saw that there wasn't, the retest reliability of one person taking the same dose was pretty solid. I mean, there wasn't a statistically significant difference, but if you look at the variability, you'll see there is still variability in terms of individuals taking the same dose twice. And again, as Stefanie and Dr. Z mentioned before, it could be because of dietary factors or other factors influencing this. As we've seen in the literature, taking rapamycin with a fatty meal can increase levels up to 30%. I think Krista shared with us, taking curcumin can reduce the bioavailability of rapamycin by up to 70%. So there's all sorts of things that are influencing these levels, which just has us look deeper into the nuances of how to really optimize dosage and regimens. And this is, the other opportunity we had where we could see, when someone takes a higher dose of rapamycin across time, does it lead to a reliable increase? And, it does increase in some, again, not as much in others. And these other factors could be influencing, these other lifestyle factors, ethnicity, other things can be influencing how much, you're able to absorb across time as you increase dose.
One thing that's important about that last slide was, there's a concern that if you take it once a week, it'll kind of build up. I think the fact that we had kind of two different samples across different doses across different times makes that a lot less likely. That you're building it up and building it up, or building some kind of like tolerance or some sort of mechanism to break it down more quickly. So this is actually pretty good.
Yeah, and like based on the literature, like extensive literature review, it seems like even taking the 6 milligram to 8 milligram dose, limited information in healthy individuals, but after a week, there doesn't seem to be detectable levels in the majority of people. So that build-up effect, especially if you're on the weekly regimen, you know, yes, ending Dr. Z on doesn't seem to be a big thing. So uh, the best practice really is routine measurement of blood rapamycin levels, at least as you're starting to take it, but also, periodically as you increase your dose and throughout the titration schedule. I think it's going to be really important moving forward.
Girish, what does the literature say in terms of what should be a reasonable target 24-hour level?
So it depends on the dose, but for individuals taking 6 milligrams per week, it seems to land somewhere around the 4 nanogram per ml range. I don't think, in Joan Manick's clinical data, she saw somewhere around the 3 nanogram per ml blood levels, and that's where you see, and that's connected to efficacy data. So that's where you see these immune boosting effects or immune enhancing effects. So somewhere around there seems to be the sweet spot from these limited studies that have actually dug into it in healthy individuals.
Yeah, because I mean, I think that's important to highlight because we're not here for this study anyway. We're not claiming that we know that answer. There are some hints from other studies, like you mentioned, but I think that the main point to highlight here is it's important to check to at least make sure you're getting some. But right now, we think the target dose or the target level at 24 hours between 2 and 4. Is that correct?
Yeah, that's what the
And
early literature seems to
yeah,
suggest.
you probably don't want to go any higher than 10. Otherwise, you start getting potentially, side effects and immune suppressive properties and things like that. So I know there's a lot of discussions on the, on the forums and things like that about taking grapefruit juice or ketoconazole or something like that. And I would discourage that because it adds like so much more noise to an already potentially noisy system. And it could be potentially playing with fire until we can understand the relationship between levels and outcomes better.
Yeah, and I think I'll just point out one before we go into. I think we can go to Q&A, honestly, to create more time for that. But and I'll put up the future steps slides. But I just want to point out with the generic rapamycin, there seems to be this overshoot effect where you're taking 6 milligram dose and you're overshooting 6 nanograms per ml and you're getting to 8 nanograms per ml at some points. We don't see that with compounded. There seems to be a buffer there. So that's another interesting point in terms of as we start thinking about side effects and who may be experiencing them and who may not. It's important to consider that as well, especially in terms of the context of measuring levels.
Yeah. As we do the Q&A, let's just stick to slide 27, the next slide, I guess.
Oh yeah, sure.
Yeah, because I'm sure this will be some of the questions we'll be referring to.
Yep.
All right, so now we did have some questions, I think, from from people who submitted them before time. You want to shoot those off and we can try to get to them?
Yeah, absolutely.
Okay.
So I have some questions here. Some of them are from Krister. So one of them is, should we strive for some kind of standardized way of sharing dose regimen between, because bioavailability differs between generic and compounded rapamycin. And the second part is, the interest in how does all this data tie into what we found with it or our activities with the Pearl study. We are using compounded rapamycin within the Pearl study. It's important, presumably, to let it be known and label that so people can start distinguishing as we move forward between the results we're getting from compounded and generic. How do this bioavailability study sort of influence the way you look at the Pearl study and the context there?
Yeah, so I mean, again, this is why it was important to have this webinar before the Pearl webinar, so we can kind of like talk about the nuances here. So for the Pearl, I think I mentioned we ended up with a 5 milligram once a week, a 10 milligram once a week, and a placebo. So based on this bioavailability data, the 5 milligram group is actually like 1.3 milligrams, a generic or commercially available equivalents. And then the 10 milligram is closer to like a 3.5 milligram or so. So even like so when we discussed the Pearl data, you know, you have to kind of look at it in the lens of potentially subtherapeutic doses. Again, we didn't know that going into it, but like a third of the way into enrollment, we have to make a choice whether to continue and hope for the best or to like scrap the whole project. So we continued hoping for the best, and then luckily this data supported the fact. And then, you know, as we talk about the Pearl data, you know, like I said, I don't want to steal that webinar's thunder. But Christopher also asked about, you know, standardizing dosings. Again, I mean, that's kind of where I'm hinting to, like, you know, checking a 24-hour blood test, and seeing if you're somewhere in that range. Like I said, we don't know what a person's optimal level is right now, and it's very likely somebody's optimal level might be different. Like let's say I get 2 nanograms and Girish gets 4 nanograms. So 2 could be good for me, and Girish could be 4 could be good for him. So it's really premature right now for anybody to claim that we know that answer. But it's important that we kind of started somewhere. And then let's see, there was a question about everolimus, which we brought up. You know, everolimus is you know, when I looked it up, it's a very expensive medication for one. It's multiple times more expensive. It is generic, but even the generic is multiple times more expensive. I think, you know, they do offer a 5 milligram tablet, but I think 4 tablets is like $500. It's like $100, you know, $120 a tablet, which makes it, you know, infeasible to either study, you know, in a limited basis like this, or from a clinical perspective. The other thing is, like, you know, all the data is on rapamycin, you know, all the, you know, the animal data and all the rest of that's all on rapamycin. So I think at this point, you know, everolimus potentially has some benefits over rapamycin, but I'm not willing to endorse it at this point until we have more human data and more efficacy data. Maybe something to study in the future, but right now I don't see myself endorsing everolimus, you know, anytime in the near future. And then any other questions, Girish?
Yeah,
Questions?
there's a few more.
Yeah.
So one of them was, did we, you know, enterically coat the capsules, or were they just the regular ones? I think we may have covered that earlier on, but
yeah, no. So the compounded capsules were just, you know, standard, anytime you compound a capsule, typically, unless you ask for a specific coating or something like that, usually the pharmacy would have, you know, like a cellulose capsule and then some kind of inert filler, excipient, sometimes they call it, and then they would have the active ingredient within that kind of that inert excipient or filler. So no, there was no attempt, like I said, because at the time it wasn't known to us about the bioavailability issue. Now, I will say we did investigate with one of our compounding pharmacy partners how to enhance the absorption of sirolimus within the context of compounding. But we kind of ditched that effort when it was found that it was more expensive than it was worth. So I think we were able to, like, I don't remember the numbers off the top of my head, maybe double the bioavailability or something of Rapamycin. So get it closer to the generic, but that more than double the cost of it. And with you know, anybody who knows the compounding space, well, a lot of times the medication or the cost of the compounding is actually in the labor required, not the drug itself typically. So we decided it just wasn't worth pursuing the additional cost of the enhanced bioavailability for compounded, because you know, in this case it just seems like you make up for it by just tripling the dose and increasing the dose. The other thing that we didn't talk about was it's potentially beneficial to have a lower bioavailability. So as long as you get enough in the blood, you know, there is a case to be made that getting some into the gut could be a beneficial thing. We don't know that yet. We did do some microbiome studies for our Pearl trial, but the data hasn't been analyzed fully yet. We're waiting for our partner, our lab partner there to analyze the data. And I suspect we would probably need larger data sets to answer that question. So yeah, we did look into enhancing our bioavailability even with the compounded, but decided against it ultimately for practical and cost reasons.
yeah, awesome. And I think I'm going to group these 3 questions together. from Round Rock Anonymous and Dayang Chen. So the question is, was data collected on subjects taking rapamycin with or without food? And, kind of how is absorption increased or affected after eating? And, the third part is, do we know of any other supplements that may influence rapamycin bioavailability? I know for that first question, we didn't specify whether a participant should take it with or without food, but any other additional insights on how food or supplements may influence bioavailability or where we're at with that?
No, I mean, we honestly, and we kind of purposely avoided coaching people on this because we wanted to kind of mimic a real world data set as much as possible. And like I said, it wasn't really known at the time. So, we were worried that if we told people to take it with food, then that could throw off our numbers. If we told them to take it without food, again, that threw it off in the other way. So I suppose like if somebody wanted to answer that, they could, take a group of people and have them take it with food and without. But then you get into the whole thing, what food? What percent fat, what composition. So you start throwing in all these variables. I think one thing we can conclude is because people, it seems like regardless of whether we told them to take it with food or not, it seems like we still got a lot of people within the same range. So one can kind of reasonably conclude that doesn't seem to matter that much.
Yeah, and I think, I mean, unless I'm missing something, those are the majority. As you mentioned before, there was a bunch of questions on everolimus and comparing that with sirolimus in terms of effectiveness and bioavailability. And I think it's safe to say it's fairly early days to make such comparisons, but interesting questions nonetheless, especially Dr. Z, as a point you brought up about cost effectiveness and accessibility, as that's a major, mission here.
No, definitely. And then there was a question about correlating side effects or maybe even outcomes with levels. I mean, that's something we're trying to do. So everybody who comes through our telemedicine platform, we're asking them to do sirolimus levels. But we're not coaching them right now. We're not coaching them on when to do the blood level. So we're getting a large amount of random data, meaning like, different time points and things like that. So it's going to be a little bit noisier data, but it's going to be a much larger quantity because, we ask for blood work, a few times within the first year and then every 6 months thereafter. So we're going to have this longitudinal ongoing data set with sirolimus levels, and then hopefully, pull from our EMR records because we ask about side effects, we ask about benefits. We monitor their dosing and things like that. So we are at some point going to have some insights, I hope, if the data turns out to be high enough quality to start making some of those conclusions. We won't know until we pull the data, but it's definitely the reason why we're measuring sirolimus levels on people. What will come of it? Only the data will tell. Only time will tell, and the data will tell.
Yeah, it's so exciting to be collecting that data and actually you're kind of having the fingers on the pulse of it. And, obviously a big thanks to everyone participating and contributing that data to make it possible, because I think we can, really make we can really move towards more personalized insights in terms of prescribing rapamycin moving forward and realizing its benefits. Are there any other of we have it listed up here, what the ongoing projects are.
So kind of like, we have a lot going on, and, of course, there's always more to do. Like I mentioned, we are requesting sirolimus levels for all of our rapamycin users. So it's going to be a much larger data set. By the way, one of the reasons why we kind of limited it to about 70 patients is because we asked a statistician, like, what's the minimum amount of data we need, to kind of reach power. And I think that was the number that came up, I think about 100 samples or so. And so we kind of, for the sake of being able to publish it, we just kind of cut it off at like 100, like roughly about 100 samples so that we can move forward. So it was, obviously we could have kept collecting more and more data, but at some point you would have gotten diminishing returns. So that's if you're wondering, if anybody out there is wondering why we kind of kept it to that number, because that was based on our statistician recommendation. And also we just like I said, we wanted to cut off and move forward. So, but there is a much larger data set that we're sitting on right now. We have not pulled from, and we hope to be able to look at that. Evaluating, rapamycin on health span. We have some surveys and things like that. that we're offering. That, we did that quality of life paper on using like the SF-36 and other quality of life tools for health span. So that is an effort that we're doing as well. Pearl trial. I mean, we have the Pearl results coming up, and then our telemedicine users are kind of like our Pearl phase 2, almost like our Pearl 2.0 sort of. So anybody who participates on our telemedicine platform will also kind of, if they consent to provide their data, of course, then we'll include it in the analysis. And then biobanking. We're reaching out to various different lab partners, to get more longitudinal, observational. So we're trying to catch people when they, before they start rapamycin, and then a few markers or a few time points after that to start gathering larger amounts of biomarker data. Because what we're collecting right now is like a quality of service, collecting kind of subjective data and kind of required blood work, blood counts, chemistry panels, things like that, lipid panels, things like that. But that's very limited in terms of looking at the hallmarks of aging or health span metrics. So we're trying to expand that into like metabolomics or microbiome and things like that. So that's kind of something we have up and coming soon. And then also, I mentioned more and more anecdotal reports, and I could have swore I heard like from 3 different people across 3 different conversations across like the same month, that people using rapamycin, it helped them either lose weight or keep it off more easily. I don't think it's a potent weight loss medication, because most people who do report losing weight on rapamycin seems to be there around the 5 to 10 pounds. So not a huge weight loss, but as somebody ages, I'm sure it becomes harder and harder to lose that weight. So we are potentially looking at rapamycin as a weight loss management tool. If for people who have lost weight, let's say on semaglutide or something like that, what it would look like, what could rapamycin, plus or minus metformin, be a tool to help people kind of keep off their weight, and then part of our observational trials to kind of track. Weight is one of the things we do track on our telemedicine patients. So we will have more of that data over time as well. Yeah,
Awesome.
lots to do. Yeah, always more to do than there's time or budget for.
Absolutely. And
Yeah.
of course,
One last thing I'd like to end with is we are modifying or changing our rapamycin. So if you go to our telemedicine platform right now, you'll see like I think it's currently listed at $149 a month. And that includes, includes everything. There's no extra cost to that. But we are making some changes to kind of optimize the pricing on it. So the pricing will come down in the near future, like the very near future, meaning within the next like week or two. With the reintroduction of the compounded offering, as well as some pricing optimization that we're doing with our generic our pharmacy partners that are dispensing the generic as well. So again, in our ongoing effort to make longevity as accessible as possible, I'm happy to introduce that we are making some pricing optimizations soon, really soon.
Awesome. And if anybody has any questions or feedback, you can definitely reach out to research at AgelessRx.
Okay, and we'll see you all at the August 27 webinar.
Can't wait.
All right.